So next we have the first of our spotlight talks, for which we have Eve Richardson, a postdoc at La Jolla Institute for Immunology, previously of the Oxford Protein Informatics Group. And she'll be speaking about machine learning, she'll be speaking about methods for antigen specificity prediction applied to BCR sequence data.
Great, thank you for that introduction. Am I sufficiently audible? Okay, good. Just check the time. So today I'm going to be speaking to you about work that's in progress for predicting BCR clone expansion following vaccination, and a specific subset of this work, which is using independent antibody sort of monoclonal databases and training models on those to do that sort of prediction. So yeah, that's our central question. Can we predict BCR clones that are expanded after vaccination? So the context for this work is that more broadly, the Peters Lab is involved in these sort of community challenges for predicting various immunological readouts following vaccination. So yeah, that's the context for this work, both in terms of how we're thinking about it, but also in terms of our cohort. So we have 19 adults who received a booster vaccination, the Tdap vaccine. So for the uninitiated, that contains antigens from tetanus, diphtheria, and the acellular pertussis, so various pertussis antigens. We just did, we have bulk, just heavy chain sequencing, just to be clear on that we did with Takara Bio using their human SmartSeq kit with UMIs. And this is all sequencing from bulk PBMCs. And we sequenced people at day 0, which is our baseline, and then we sequenced them at day 7, which corresponded to the antibody secreting cell peak in the accompanying cell frequency data. So the way we've currently formalized this prediction task is to have our negatives as everything that we see at day 0, and then to try and isolate the most interesting and most likely to be actually antigen-specific clones that we observe after vaccination as taking a particular subset of day 7. And clearly, how you define what's expanded is not a closed question, so we explore various different thresholds for defining that expansion. And I should also say here that all of the data is subset just to the IgG, IgA, and IgE reads, because a model doesn't have to be very clever to predict class switching. So this is, you end up with this collection of data, a collection of BCR sequences from day 0, day 7, and removing the intersection of reads, a very small intersection of reads, of clones rather, that were found at both time points. And then we modulate how expanded a clone has to be in order to be a positive label. So for example, if we took the top 1% of clones per subject, as we define it on a per subject basis, we'd get about a 1 to 99 positive to negative imbalance with about 76,000 clones in total. And so this work is one way of doing this. So just to be clear, we're not training on this sequencing data. We're training models on completely independent monoclonal antibody databases and applying it to the sequencing data. And we have a reason for thinking that this might work. Substantial, lots of previous work has shown that you can use databases of previously described antibodies either to epitopes like you get in the IEDB, and there are many other of these antibody databases to particular antigens of interest, some of which we've used here. And then use some similarity metric. So clonotyping has always been, I think, a very sensible thing to do. And work from Genentech in 2021, as well as our work in the same year, showed that clones that you pick out from heavy chain sequencing that are clonally related to your database, there's quite high confidence for antigen specificity. But we also show in this work, which is unfortunately under review still, but hopefully will be out in the near future, showed that this method applied in the context of Ebola vaccination was highly correlated with the expanded clones and was also correlated with antibody titer to the vaccine antigens. And so this was our first step, was to create a database of tetanus and pertussis monoclonal antibodies. So we collected those from the literature. A special thank you to Jake Golson for having shared many tetanus antibodies with me, actually years ago at the beginning of my PhD, which became quite helpful, as well as some pertussis antibodies, including ones that we had discovered in the Kai mouse a few years previously. So ending up with around 600 antibodies in our monoclonal database. And we use a search technique called clone search, and that identifies clones that have the same IGHV and gives you a CDRH3 sequence identity to anything in your database. So we use a cutoff of 70%, as in previous work, and we could see from our data that that clones that that had similarity to our database were significantly expanded relative to clones that were not. And to formalize that in the prediction, in sort of the prediction sense, we describe this in terms of a ROC AUC and an early retrieval ROC AUC, using that score as the prediction, and then the label being expansion at some level. So, for example, if you use a 1% clone frequency as your criteria for positive versus negative, you get a ROC AUC of 0.73. So there's already, quite, I think, powerful prediction in the sequence identity baselines, as you will see. The question is, can we do better than sequence identity? So we've all seen so many antibody language models and protein language models that have been developed in the past couple of years. And then there's also inspiration that we can take from TCRs. So the IMREP challenge, for example, evaluated many of these model architectures that we can repurpose maybe for BCRs. So the first step to train these models was to do our database curation. As I described, we had built this database of tetanus and pertussis toxoid antibodies, and we supplemented that with negative data from the IEDB, from Covabdab, and from an Ebola virus antibody database called Ebovabdab. And for lots of this data, some of this data goes back early 90s, where all that was reported was IGHV genes and CDRH3s. So to sort of expand the possible space of featureizations, we did this processing. And we wanted to benchmark a large variety of models using different featureizations and architectures, so including using the IGHV, so using embedding layers for that, or just a CDRH3, using protein language model representations, the full heavy chain sequence, as well as various CDR models. So yeah, we tried to cover a good diversity of architectures and featureizations. And then we compared that to sequence identity baselines, clone search, as I described, but also a simple KNN on CDR identity. So first of all, we wanted to check that our models were doing something sensible. So we trained and tested these on data published with DeepAir that many of you will be familiar with, and part of that was this BCR classification task. So the long and short of this is that we were able to, with at least one model or multiple models in our benchmark suite, achieve parity or sometimes slightly better performance versus DeepAir. The final column is our top model across all benchmarks. So basically our model fitting was sensible. And then we applied it to our own database. And so these are the results from cross-validation, which we did in order to do the hyperparameter selection. So you can see that over the full ROC curve, we have significant improvements in classification performance versus the sequence identity baseline, less so in the early retrieval AUC. So then we used that for our selection of hyperparameters, and then trained that and then tested that on our holdout test set. So you can see, again, there's a clear performance increase in the total ROC AUC but that in the early retrieval ROC AUC, the sequence identity baselines are quite hard to beat, and that's why it's always important to include them. And finally, we decided to apply those models to the BCR sequencing data. And the way we currently have it set up is that you make a prediction per sequence. Those sequences can then be grouped into clonotypes. So I should have said that our clonotype definition is IGHV gene matching, V and J gene matching, and 90% nucleotide identity, so quite a standard clonotype definition. But then you get this set of scores, and you have to come up with some way of combining those scores to produce a final clonotype score that you can compare to your label. And this is, the last slide. So this is the performance using different types of pooling, how you take the scores from the clonotype and compare that to the label. And the x-axis is how stringently you define clonal expansion. So on the left-hand most side, that would be how well can you identify the most expanded clone after vaccination, all the way down to how can you identify the top 10% versus what you sequence at day 0. So you can see that with maximum pooling, you have significant increases in the AUC that's achievable versus a sequence identity baseline. And median pooling is more sort of punitive of the performance. You can see there's a general decrease overall. And we're sort of finding some way to what would probably be a happy medium between these two. The max pooling is not the most principled approach. So we will in future be sort of exploring different clonal definitions to try and sort of reduce this difference between our performance depending on our pooling. So in conclusion, I would say this because I work at the IEDB, but public antigen and epitope-specific antibody databases are really crucial tools for interpreting BCR sequencing data, and we should try to democratize those as far as possible. I think we've shown that in bulk BCR sequencing data, you can train models on these databases to be predictive of clone expansion in response to vaccination in completely independent databases. And finally, I swear I wrote this prior to the machine learning working group announcement, but yeah, there are no established benchmarks for things like antigen specificity prediction, tasks like this, clone expansion prediction, and we're really excited at the prospect of those becoming community challenges where hopefully our benchmark suite can be a starting point. So with that, I'd like to thank everyone in the Peters Lab at the La Jolla Institute for Immunology, the CMIPB team, IEDB team, and the LJI sequencing core. My poster was yesterday, but come and speak to me if we're interested, and there'll be a poster on the CMIPB work tomorrow that Bjorn will be presenting. So yes, please do get in contact, and I'm happy to take any questions. Thank you.
Well, thanks, Eve, and thanks for that plug for the machine learning working group. Great. We've got a question come through from the Slido. Would you want to control or downsample your number of clones per sample to better identify the expanded clones, or search across as much data as possible here?
Yeah, that's a really interesting point, because there is definitely subject variability in terms of the number of expanded clones per sample. So we try to control as much by defining the expansion on a per subject basis, but that's definitely something that we will, I think, be looking into.
Great. And finally, do you think predicted structural similarity methods could also be worth looking at for predicting the expanded clones?
Yeah, I think that's definitely. Clearly there's a body of work demonstrating with tools like SPACE and SPACE2 that you can use stuff like CDR-MSD to group related antibodies. I would be interesting whether that could hold with so many more antibodies, many of which are non-antigen specific. But yeah, in the future, I think that's definitely an interesting route. Thank you. Oh,
we've got a last second. Does prediction capacity differ by epitope, antigen, virus based on the database size?
More generally, applying this, certainly it does. I suppose I don't know yet sort of the parameters of how prediction performance would vary according to those things. Certainly this was, I suppose, quite challenging compared to other targets where there's really not that many tetanus or pertussis antibodies, at least publicly available. But this is really an interesting question.
Fantastic. Thanks again, Eve. So for our final talk of the session, we have Catherine Wong, a senior scientist at Roche Diagnostics, who will be giving a presentation on leveraging large language models to enrich clone selection and characterization in antibody discovery.
Good afternoon, everyone. Sorry about the delay. So first of all, thank you to the organizers for putting together this meeting and for offering me the opportunity to stand here and talk to you about our work. And secondly, also thank you to all my friendly colleagues listed here all across from immunization, high-throughput screening, and machine learning. Without them, I won't have this wonderful data set to play with. So the idea of this presentation is not to show you any groundbreakingly new machine learning method, but to show you two data sets that we have and what we have tried with large language model that seem to have worked, and some of our perspectives on the kind of sequencing data that we have based on the latest advances. So I'll get started with the first data set. So the aim of this data set is actually to compare the clones selected from the traditional clone picking, so B-cell cultivation, and from single-cell sequencing workflows. So first off, we started with 3 animals, and during the immunization, we took 2 bleeds and sent them for screening for the clones that have the desired phenotype, so binding characteristics. We take them out and send them for sequencing. So in this way, we have the sequence phenotype mapping, but only a very small number of them. And in a single-cell sequencing workflow, in the same rabbits and in the same immunization, we took 5 bleeds, 1 bleed before immunization and 4 bleeds during the immunization. And one of the bleed is Fax-sorted on the antigen, so we expect a high proportion of real binders in that bleed. And then we send them off for single-cell sequencing, and in this case, we're using the 10x platform. We don't know about the phenotype of all of these sequences, and we don't even know whether all of them are binders. But what we do have is a lot more sequences than what we get from the clone picking workflow. So we wonder then if we can use large language models to help us understand this data set a bit better and how that compares with the classical sequence-based clustering. So we train our large language model in-house with no refinement on this particular immunization data set, and then take the embedding and put on t-SNE projection to get this visualization. So the coordinates from this plot, they're from the t-SNE projection. And for visualization purposes, I only show you the top 20 largest sequence families based on sequence-based clustering. And what we see is that members from the same sequence families tend to sit around a similar embedding space, even though the embedding space also sometimes subdivided these sequence families into smaller subclusters. And so based on this, we think that on a very broad brush way, large language model embeddings can capture some of the sequence relatedness, but also give us an orthogonal metric to the classical sequence-based clustering. So the second question we asked is if we have if the idea of embedding space works, can we see the overlap between the clone picking workflow and the repertoire sequences? And here I'm showing the picked clones in crosses, and roughly they do overlap with, they do cover, the repertoires did cover the majority of them, and in fact, depending on how strict you define your sequence relatedness or clustering metric, we did recover 70 to 90% of the picked clones in the repertoire sequences. And the third question, if the idea of embedding space works, does it mean anything functionally? So since we have the coordinates for and the phenotype of the subset of clones from the B-cell cultivation workflows of the picked clones. I'm now coloring the crosses by the phenotypes. So very roughly speaking, again, if we have clones that are sitting in the neighborhood in the metic space and also coming from the same sequence family, they tend to have the same color crosses, meaning that they have the same phenotype. And the fourth question that I have, based on this data set, because we have a tracking of the immunity development, is that we wonder if we can predict which bleed a sequence comes from. Originally, we were trying to restrict ourselves to sequences that are related to the binders. That is, the sequence families that have at least one clone from the antigen-sorted bleed. And based on that, we use very basic one-hot encoding, and then the large language model embedding, plugging on random forest LSTM. That didn't work at all for the prediction, but then even just a simple neural network that gives us a good test accuracy of 87%. When we take a step back and use the entire data set, we still keep a test accuracy of around 80%. So at this point, we think that large language model can tell us a bit more about the functional, or at least contain more information about the functions of these antibodies, and can potentially be complementary to the sequence-based clustering. So moving very swiftly onto the second data set, this time I'm not going to show you any machine learning-based results because we are still refining it, and I wanted to test on a different data set before showing it to you. So in this data set, we have not just the antibody sequences, but also which antigen they bind to, and in one run we can put in up to 16 antigens in one mix. The hope is that we can complement or even replace the clone picking workflow, and with a greater sequencing depth and also the extended antigen information. As a side product, sort of, we also have the single-cell gene expression data of all of these B cells, and on the left you have the classical UMAP of the gene expression colored by Leiden clustering. What was quite interesting is on the plot on the right. In this experiment, we have 4 animals, 2 transgenic animals against one antigen, and 2 wild-type animals against a different antigen. So here we see a good separation of not just the genotype, but also, if you consider that way, also the antigen reactivity. So we wonder if we can use the gene expression to help us understand this, or to help us pick clones better, essentially. So we did. So we asked the question of whether we can identify genes that indicate the response to certain antigens. And so in this case, we run the differential gene expression analysis, and we saw that there are genes that are expressed universally across all the B cells. There are marker genes for expression, for expansion, for survival, for maturity, but also marker genes that are differentially expressed when the animals are challenged with different antigens. So at this point, we think that with the gene expression data, with the antigen information, and also various representation of the antibodies from sequences, structures, and language model embeddings, we can have a better model to help us with selecting antibodies for the discovery workflow that we have. So by way of very brief summary, sorry about the formatting. So I've shown you 2 kinds of data sets we have, and some of the tasks we asked language model embeddings to try to do. That seems to have worked. We think that large language model embeddings can contain more information than just sequence-based approaches, and based on and given the advances in sequencing technologies, we're able to know more about the B cells itself, and that can pave ways for multimodal modeling, as we see a lot in TCR analysis. So my poster will be tomorrow. If you have any better ideas how to use and analyze this data set, to come to me. I'm the second last poster. So with that, I'd like to thank everyone for listening.
Thanks, Catherine. We've had a few questions come through on the Slido, if you wouldn't mind. First one, might have missed it. What was the encoding used? Were sequence features grouped separately while encoding? Do you think this makes a difference?
I might not have understood the question correctly, but we're just using what we train in a BERT-based language model we trained in-house and took out the embedding once we run through our sequences in it.
Okay,
So
an easy
that's-
one next. Why TSNE instead of UMAP?
Yeah, the UMAP one, somehow there was some numerical instability, I don't know why. The UMAP looks like having some spaghetti on the outside, so it's not a good projection for our, I would say, short encoding factor.
We always save the controversy for the end of the day. Do you think adversarial training would make a difference?
This is actually something we had explored. You have to be very careful with your, your definition. That's all that I can say.
Okay. Finally, is BEAM part of your standard workflow for antibody design or more experimental?
Yeah, so BEAM, we've actually just established it in February, so I wouldn't say that we have put it into regular use yet. Yeah, I think that's good.
Great. All right, well, thanks again, Catherine.
Thank you.